And my name is Samantha Knott from Immuto Scientific, located in Madison, Wisconsin. I'd like to thank the session chair and the ASMS organizers for giving us the opportunity to demonstrate and show our exciting work, complementary trifluoro and hydroxyl radical labeling, a powerful tool for rapid protein footprinting. So understanding the way proteins interact with the world around us is not only important for drug development, but more generally the pharmaceutical industry. So here on the left, I have a classic example of a monoclonal antibody binding to its antigen. And mapping the precise site or set of amino acids where that antigen binds to the antibody is called epitope mapping. And better understanding epitopes is extremely important to help us develop and aid in more potent drugs, and also may provide and help provide patent protection and potentially protect a product if any case arises. Importantly here, 3D structure is very important, meaning mapping conformational epitopes or a discontinuous set of amino acids where the antigen requires a tertiary structure to bind often provides much stronger patent information and data than mapping of a linear epitope or a continuous set of amino acids. And the monoclonal antibody industry is $100 billion. Pharmaceutical industries have a landscape of technologies to choose from, and every technology should be thinking about managing or balancing resolution and throughput. So techniques like X-ray crystallography and cryo-EM provide atomic level resolution. They're excellent. However, they tend to be low throughput and cost ineffective. On the other hand, really high throughput techniques like phage display tend to not yield or give the conformational three-dimensional epitopes that we're looking for. Striking a healthy balance between these two is covalent labeling coupled to mass spectrometry. So why will R here? And in particular, hydroxyl radical footprinting is using hydroxyl radicals to label the solvent accessible surface area of a protein. And I want to walk through exactly how this workflow works for people who may be unfamiliar. And the first step is we have a protein in solution, and we have some sort of method of labeling the solvent accessible surface area with those hydroxyl radicals. We then quench any unreacted radicals and digest our protein. And importantly, here you see some of those peptides have that label on them, some of them do not. We then use LCMS and data analysis to identify and also localize where that modification is. Here at Immuto, we use protein metric software, works very well. We can then take an extracted ion chromatogram corresponding to both the unmodified peptide and the modified peptide. Once we take the area under that chromatogram, we can look at the percent modification of any individual peptide. And importantly, we can look at two different states. So for instance, apo versus holomyoglobin, with or without the ligand. Or you could do an antibody with or without its antigen. And here the percent modification is taken as the modified peptide area over the modified plus the unmodified peptide area. Now, if we look at percent of significantly changed peptides or regions, it's going to be indicating to us there may be ligand binding, or it may be indicating that that is the epitope of the antibody and antigen. And there are a variety of ways to do this. We've talked about FOX already today. Here at Immuto, we use plasma-induced modification of biomolecules. So here's a diorama of our setup. And importantly, here we have a high voltage electrode, and that electrode generates plasma. So this plasma is high energy electrons and particles, and these are creating our hydroxyl radicals in solution from water. Those hydroxyl radicals will label that outside or the solvent accessible protein with the hydroxyl radical. And we also have a radical dose measurement and feedback system, so we can do this really accurately and consistently over time. Importantly, this is happening on a sub-microsecond time scale, so very, very quickly to prevent any unfolding of the protein. I wanted to show that we can make this 96-volt plate compatible, so we can automate this and have done so in our lab, and also show an up-close picture of the plasma because it's really neat to see. And people have been doing this for a while and have observed that we can label all 20 amino acids with a hydroxyl radical at the physiological pH. However, some residues like cysteine or tyrosine tend to be much more reactive and are labeled much more efficiently. Whereas some residues like glycine and alanine are labeled at a much lower rate, and we don't see them labeled as much as we'd like to. An example of this is standard protein myoglobin. And as you can see here, we have OH labeling. And I didn't mention this yet, but we also observed nitration or NO2 labeling of our proteins from nitrogen in the air. And if you zoom in on any area of this myoglobin there, you can see that there's some areas where we don't observe any blue, or we didn't observe any label there. However, we may want a label there or need a label there. So there have been several groups, including the SHARP, the Gross Lab, and they've been really pushing towards introducing new chemistries. So introducing new reagents so that we can better label those less reactive residues. And in this case, these groups have used Van Gogh's reagent, which uses hydroxyl radicals to generate a CF3 radical in solution that can then label solvent-accessible residues. And the standard system, which we wanted to start with and see if we could apply it with our PLIM system, is looking at apo and holo myoglobin, so with or without its ligands. As you can see here, our workflow involved taking myoglobin with our Van Gogh's reagent and labeling the solvent-accessible surface area with OH and CF3. We then digest and use LCMS to identify and extract and quantify. Here's our first set of results with myoglobin. As you can see here, we were really excited to see that we have a lot of residues that are preferentially labeled by CF3, including alanine and glycine, those less reactive residues. We have some residues where we observe labeling by both species, and then some that are only labeled with our standard PLIM system. If we combine the information or both labels, we go from labeling about 14% of the protein to 60% of the protein. And if we zoom into that same area, we now see a label on almost every single one of those residues. Now, we wanted to extend this to an actual drug antibody interaction. So we were looking at HER2 and Herceptin because it's also really well characterized, and seeing what kind of information we can gain. So the workflow is similar where we have a solution of protein, and then we have a sample where we have HER2, and we have a sample where we have HER2 plus the antibody, and we label both of them in solution. We then digest and do LCMS, and we use data analysis and validation to look at the percent modification with or without that antibody present to bind to its antigen. We then can use that data to see areas where there's a significant change, which may be indicating a structural or an epitope region. And here is the data we got from our HER2 and HER2 plus Herceptin. These were the three most changed or significantly changed peptides we observed. And as you can see here, we observed protection where we see less modification upon antibody binding. We also see deprotection, which often can happen when it's really close to the region, because that structure will sort of pop out in solution and become labeled a little bit more. And when we align these peptides to cryo-EM data, or the crystal structure that's already been obtained, it aligns identically to the epitope that has already been mapped, which is really exciting. Now, what can CF3 add? So we repeated this experiment, and we just added our Van Gogh's reagent to generate CF3 radicals. And here's the data we saw. So while we primarily observed cysteine, phenylalanine labeled with OH labeling alone, when we add our Van Gogh's reagent, we were able to label every single one of the amino acids, all 20, which is really exciting. And whereas we had a label approximately every 6 amino acids without the Langois reagent. With it, we now have a label every 1.3 residues, or approximately 50% of the protein is labeled. If we zoom in on any one of these epitope peptides and look at residue level information, this is also really clear. So with OH labeling alone, we're labeling cysteine, histidine, and we observe changes in their level of protection or modification. When we look at what we get when we add our Langois reagent is we now have 8 residues continuously covered where we can monitor and probe the amount of modification across the entire epitope region, which is great. And what we saw was that this alanine and cysteine in particular were significantly changed, indicating they might play a bigger role in that epitope or drug antigen interaction. We looked at our other epitope peptide and observed a very similar situation where we only labeled 2 residues on that peptide with OH alone. And when we add our CF3 reagent, we now are able to label 5 different residues and monitor them in solution. And we saw in this case, the cysteine, valine, and alanine were significantly changed and may play a bigger role once again. So in summary, CF3 labeling really improved our coverage in our standard hydroxyl radical footprinting workflow with PLIM of both myoglobin and HER2. And the resolution of our epitope mapping data was significantly improved because it provides both a unique and complementary radical label that we can look at. So what's next at Immuto? At Immuto, we're now trying to integrate the CF3 labeling into our standard workflow for epitope mapping. It gives us more information, and we're going to continue to try to label more difficult proteins like multi-pass transmembrane and integral membrane proteins. Lastly, we're continuing to work on our automation so that we can do this for high throughput screening and have a fast turnaround for any clients. And I'd like to thank CEO and CTO, Faraz and Dan, for being really helpful in making this work possible, as well as James, our Director of Technology, and the rest of our board here at Immuto. And lastly, we'd like to thank Protein Metrics for giving us the software capabilities so that we can make this work happen. Thank you.